Can you tell me what's the benefit of seeing the DNA inside a nucleus?
So in 1953, Watson and Crick discovered that DNA is a double helix. And that structure really revealed how our genetic information is stored and copied. But in the cell nucleus, 2 meters of DNA has to get compacted into a structure that's 1/millionth of a millimeter. And so the structure, DNA is not a double helix in the cell nucleus. It actually gets folded into these other structures. And we don't know what they are. We haven't seen them, right? We haven't seen these structures that determine if our genes are accessed and used, right? And structure determines function.
And you say up until now. Can you talk about your team's discovery?
Yeah, sure. So what Horn-U in my lab discovered was a DNA dye that diffuses into the cell nucleus. And when you shine light on it, it actually catalyzes a metal dusting on the surface of the DNA so that it can now be revealed in the electron microscope. It creates kind of a metal cast. And together with Mark Ellisman and colleagues at UCSD, he's created 3D imaging techniques, which is like a CT scan of a body. It's a CT scan of a cell that then allows you to reconstruct sort of the metal cast of DNA. And so for the very first time, you can see actually DNA structure inside the cell take a journey and from sort of base pairs to megabases, right, across these scales. And that's the scales at which biology occurs. That's what we've not been able to see, right? How different DNA structures actually give rise to different genes, different cells, and different cell fates.
So how does seeing a 3D image of a genome allow scientists to possibly manipulate it?
Right. No, so that's one of the most exciting things, right, is in cancer, certain genes are turned off. They're there, but they are what are called silenced. And that's what gives rise to uncontrolled replication. And viruses can do the same thing. And so the question is, could we turn these things back on, make a tumor cell remember the structure that could make it normal again? But we don't know what those structures are. So people have been trying to develop epigenetic drugs, but it's been based on really not knowing what we even were trying to change, okay? But now we can see the structure. We could actually potentially design molecules specifically to those shapes that only turn on those genes. Pathologists grade cancers based on a light microscope at 20x. We can now see it at 29,000x, and we can see the genome. So now basically what those structures will tell us could help us diagnose individual cancers and see actually that in a completely new way, but also design new drugs that specifically go after those structures that are unique to them. And that's kind of wild.
And for the layperson at home with not very much science knowledge, or the one sitting right in front of you, just to sum it up, how significant would you say this discovery is?
When you see something for the first time, the universe expands, you know. Things change, and you begin to see what a transcription factor would see, what a virus would see. And it gives you this sort of ability now to navigate, and I hope to change the realities for cancer patients in a way that we were blind to before, right? Because without illuminating those structures, you can't rationally design drugs against what you don't know and what you can't see, right? You're chasing a ghost. Well, we've taken sort of the sheet off, and now we can see underneath, and it's not as scary when you know what it is.